be discussing African population history based on patterns of genetic diversity. So why do I think it's important that we study African genetic variation? Well, for one, if we want to learn more about modern human origins, we need to be looking in Africa, which is the site of modern human speciation. Secondly, if we want to learn more about African American ancestry, this will be an important region to study. Third is that Africa is a region with a very high level of infectious disease, with HIV, malaria, and TB being three of the biggest killers, but there's also an increasing level of non-communicable diseases like diabetes, for example, and cardiovascular disease. And African populations have been greatly underrepresented in the biomedical research, and so we really need to give more focus to these populations so that we can come up with better diagnostics and better treatments for these diseases. And lastly, we know that people differ in regards to drug response, and this is likely due to variation at drug metabolizing genes. But again, we currently know very little about the extent of variation among Africans at these loci. So first, I have to give you a little bit of information about African population history. There are over 2,000 ethnic groups in Africa speaking distinct languages. And these languages have been classified into 4 different language families. So in blue are languages classified as Afro-Asiatic. They're found predominantly in the north and northeast of Africa. And these would include, for example, Semitic languages, which are also spoken in the Middle East. And they would also include Cushitic languages spoken in Northeast Africa. And then in red, we have populations that are speaking Nilo-Saharan languages. These tend to be pastoralist groups like the Maasai, for example, who live in Kenya and Tanzania. And these populations are mainly found in Central and Eastern Africa, although there are a few groups who have migrated to the west of Africa. The most broad spread language family consists of the Niger Kordofanian languages shown in yellow or orange here. And the most common subfamily is the family of Bantu languages. Now, those are thought to have originated in Cameroon or Nigeria around 5,000 years ago, together with the development of iron tool technology, and which led to much better methods for practicing agriculture. And so these populations had a technological advantage in a sense, and they were able to rapidly expand across Africa into East Africa and then South Africa, or from West Africa along the western coast into Southern Africa. The 4th language family, shown in green here, is classified as Khoisan, and it consists of languages that have click consonants. So these are found predominantly amongst the San hunter-gatherers in Southern Africa, and also amongst 2 groups called the Hadza and the Sandawe, who live in Tanzania. Now, despite the importance of studying Africa, there have been relatively few genomic studies in that region, and there's a number of reasons for that, and one of which is just the challenges of doing research in areas that sometimes have little infrastructure. And so I wanted to show you some examples of the fieldwork that we've done over the past 12 years. We've mainly been studying minority populations in Africa who practice indigenous lifestyles, and they live in very remote areas, so we have to, for example, have a 4-wheel drive vehicle. And this work has been done not only by myself, but by my students and postdocs and African collaborators over many years. So here's an example. I like this. It shows my postdocs, Alessia Ranciaro and Simon Thompson, and they were doing an expedition in Ethiopia in 2010. We basically have to bring all of our lab equipment with us. And I like this because it shows both the outside perspective of the car and also the inside perspective. These are some of the other challenges that they faced. They were there during the wet season, making it extremely challenging to travel. In each of these regions, we typically start by doing what you could think of as town hall meetings, in which we explain the research to the community, and we explain both the risks and the benefits and make sure that they understand why we're doing this research and how it might benefit or not benefit the community. Ultimately, though, we have to obtain individual informed consent to do this research. We also measured a number of phenotypes like height and weight. More recently, we've been looking at more detailed anthropometric, cardiovascular, and metabolic traits. From each of these samples, we typically obtain blood intravenously, and we've started to also obtain RNA. But one of the challenges is processing these samples in regions where there's no electricity. So here's an example where we set up the so-called bush lab. We had to set up our centrifuge and hook it up to the car battery. But in other areas, we can find a local clinic. They'll often have a generator, and so then we're able to hook up a larger centrifuge. One of the ways in which we obtain DNA, and the DNA, I should note, is only present in the white cells of blood. So the first thing we're going to do is we're going to break open all the red cells. And we do that by adding a solution that's going to cause them to burst open. Then we're going to spin down the samples in the centrifuge. And we have to repeat this several times, and we're going to end up with these little pellets at the bottom of the white cells. And that's where we're going to find the DNA. Here are some other challenges of processing in the field. After we've isolated the DNA, we add another buffer, which is going to preserve the samples at room temperature. But here's a case where Simon Thompson actually had to bring a generator with him and set up the entire lab in the bush when he was studying the Hadza hunter-gatherers of Tanzania. Another very important thing is to increase training and capacity building in Africa so that they can do this research themselves. And that's something that I've spent a lot of time doing, and I think is very important. Also equally important is actually returning results to participants. And it's really surprising how little this is done, but I can assure you that people really appreciate it when we return the results. And I think it's also an ethical obligation so that they can benefit from what we learn from these studies. So I want to start by talking about some of the phenotypic variation that we see in Africa. This is an example of skin melanin levels or skin pigmentation. So the higher the value here, the darker the skin color. And I want to just make the point that we see a lot of variation in skin pigmentation levels across diverse Africans. And one of the things that we're interested in looking at is correlations with vitamin D, for example, because we know that vitamin D is produced by UV light and that people with darker skin may produce less vitamin D, for example. And vitamin D can have important health implications, so this is relevant to know. It's also an interesting trait to look at how people have adapted to different environments. Here are the results of a principal component analysis for a number of cardiovascular traits. And these are different populations. If the populations cluster close to each other, it means that they're very similar for these traits. And we've color-coded them based on shared language and ethnicity. And what's interesting is that they tend to cluster based on language and culture. So here are the Nilo-Saharan speakers, here are the Afro-Asiatic speakers, and in yellow here are the Niger Kordofanian speakers, but we see two exceptions. These are two groups that live on the coast of Kenya in geographic proximity to the Bantu-speaking groups, suggesting that not only are genetic factors important, but environmental factors are probably quite important as well. And here we can see tremendous variation for height, weight, and BMI in Africa. And again, we're seeing that populations tend to cluster based on shared ethnicity. And at the extremes, we have the very short-statured pygmies from Central Africa. And then we have the very tall and thin individuals from Kenya and the Sudan. And so as we'll talk about in the last section of my lecture series, this may be due to adaptation to different environments. So now I want to tell you about the patterns of genetic variation and genetic structure in Africa. And this is based on a paper that we published several years ago, in which we looked at genome-wide variable markers. And these were genotyped in over 2,500 Africans from 121 ethnic groups that are shown by these dots here. But note that even though this was more than had ever been done before, it still represents just a fraction of the 2,000 ethnic groups in Africa. So we're still missing a lot of the variation. We then looked at 98 African Americans from 4 regions in the US and a comparative data set of about 1,500 non-Africans. So let me first tell you about the levels of genetic variation that we saw, and that's indicated by the height of this bar. And I've color-coded this by geographic region. So shown in orange are people from Africa, and as nearly every study has shown, Africans have the highest level of genetic variation. And then we see decreasing variation as we move west to east across Eurasia into East Asia, Oceania, and the Americas. So the patterns of genetic diversity that we're seeing simply reflect our evolutionary and demographic history. We see the highest levels of diversity in Africa, which is the site of origin of modern humans. And then when small groups of people migrated out of Africa within the past 50 to 100,000 years, there was a population bottleneck. And so we see a decrease in genetic diversity. And as humans migrated west to east across Eurasia and into the Americas and into Oceania and so on, there were a series of founding events. And again, a concomitant decrease in genetic diversity. So this is a phylogenetic tree constructed based on pairwise genetic distances between populations. You can't see any details on this tree. I just want to point out some overall trends. And I've color-coded these such that the populations shown in black with black branches are non-Africans, and then the Africans are shown here. So the first thing that you can see from this tree is that non-Africans are distinguished from Africans, and that the non-African populations are clustering by major geographic region. So we have people from India, Central Asia, Europe, Middle East, East Asia, and the Americas, and then Oceania. And even within Africa, populations are clustering by major geographic region. So here are populations from the north of Africa, from Eastern Africa, from West Central Africa, and then from Southern Africa, with one exception. Down here, at the root of this tree, we see the San hunter-gatherers from Southern Africa, but clustering near the San are the Pygmies who today live in Central Africa. And that's really intriguing and maybe telling us something about the history of these populations, and I'll discuss that more in a moment. Now we can also compare genetic distances, which are shown on the y-axis, to geographic distances between pairs of populations, shown on the x-axis. And we see a significant positive correlation. But we can also see a lot of scatter here. And what that means is that there are some populations that are geographically very close, but they're genetically very different. And those probably represent recent migration events of genetically differentiated populations. And then on the other end of the spectrum, we have some populations that are genetically very similar to each other, but geographically very far apart. And those may reflect, for example, the Bantu people who migrated from Western Africa to Eastern and Southern Africa. So they've gone quite a long geographic distance, but genetically, they're still very similar to each other. So now I want to move away from looking at populations, and I want to talk about looking at variation amongst individuals. And the first thing I want to show you is a principal component analysis based on individual genotypes. And so each of these circles actually represents a person, and if they cluster together, it means that they're genetically similar to each other. So as shown here, the first principal component accounts for as much of the variability in the data as possible, and each succeeding component accounts for as much of the remaining variability as possible. So the first principal component essentially is differentiating the African groups from the non-African groups. The second principal component is differentiating the Native Americans, Eastern Asians, and Oceanian populations from the rest of the world. And the third principal component is distinguishing the Hadza hunter-gatherers from Tanzania from the rest of the world. This next result is based on a probabilistic analysis that simultaneously infers ancestral population clusters, which are represented by the different colors shown here. And then we have, this is actually composed of a series of lines, and each line represents an individual. And an individual can have mixed ancestry from different ancestral population clusters. So what we tend to see outside of Africa, which is shown along the bottom here, is that individuals are clustering by major geographic region. So in blue, we have individuals who self-identify as European or Middle Eastern. And then here we have individuals from Southern India. Here we have individuals from Pakistan, Central Asia, East Asia, Oceania, and the Americas. But what I want you to note is all the colors that we see here in Africa. That's representing the very large amount of genetic diversity, not only within, but among African populations compared to the whole rest of the globe. I'll just point out a couple of trends. In orange colors are populations from Central and West Africa who speak Niger Kordofanian and Bantu languages. In purple are populations that speak Afroasiatic languages and originated from Northern or Northeast Africa. In red are populations that speak Nilo-Saharan languages, and they most likely originated from Southern Sudan. We have populations that are speaking Chadic languages, a group called the Fulani, who are nomadic pastoralists. Most of the North Africans have a lot of European or Middle Eastern admixture. And then we have the hunter-gatherer groups like the Hadza, the Sandawe, Pygmies from Central Africa, and the San hunter-gatherers from Southern Africa. Now, we repeated this analysis within Africa, and again we inferred 14 ancestral population clusters. But for ease of viewing, I'm just going to pool individuals together and show them as pie charts. Now, first I'm showing you the 3 populations that have been studied as part of the HapMap and 1,000 Genomes Initiative. These are NIH-funded programs to characterize genetic variation across ethnically diverse human populations and making that data publicly available so that it could be used by a wide range of biomedical research scientists. Now, what I want to point out is that when we look at the rest of Africa, we see quite a bit more variation. And so, for example, populations in East Africa look distinct from populations in Western Africa, Northern, and Southern Africa. It's also interesting because we can see remnants of historic migration events. So, for example, I mentioned to you the Bantu migration. The people who speak Niger Kordofanian or Bantu languages are represented by shades of orange. And you can actually see that they appear to have originated, as I said, in Cameroon, Nigeria region, and then they migrated across Africa into Eastern Africa, where they admixed with the indigenous populations there, and they also migrated into Southern Africa, where they admixed with the populations there. We can also see remnants of migration of individuals from Northeast Africa who speak Afroasiatic languages into Kenya and Tanzania. We see migration of people who speak Nilo-Saharan languages originating from Southern Sudan. There was one group that went west, and we think that some of these people who speak Chadic languages, which are actually classified as Afroasiatic, genetically they look very similar to the Nilo-Saharans. So in fact, there may have been a language substitution at some point in the past. And then we have migration of the Nilo-Saharans, pastoralists into Kenya and into Tanzania. We can also see that some of the hunter-gatherer groups are very distinct. Here are the Hadza hunter-gatherers who speak with a click in Tanzania. Here are the Sandawe who speak with a click, also in Tanzania, but their languages are very divergent from each other. Here are the San hunter-gatherers, shown in light green, also speaking with a click, but again, their languages are very differentiated from the other two populations who speak with clicks in Tanzania. And then we have populations from Central Africa. Interestingly, the pygmy population called Mbuti, who lives the furthest to the east, appears to possibly share some common ancestry with the San. And in fact, several pieces of data that we've studied suggest that there could have been a proto-Khoisan pygmy hunter-gatherer population in Africa that probably existed greater than 50,000 years ago, and then underwent population divergence and differentiation and then migration within the past 50,000 years. But there's still a lot of work to be done to try to differentiate this population history. So next I want to talk about what we found in terms of African American ancestry. We looked at African Americans originating from 4 regions in the US: Chicago, Pittsburgh, Baltimore, and North Carolina. Now, not surprisingly, you can see that the majority of ancestry is this Western Niger Kordofanian ancestry shown in orange. The other major component of their ancestry, which is summarized here, is European ancestry, which ranges from about 0 to greater than 50%. And then we see small amounts of ancestry from other populations, including some other African populations who speak Chadic languages, for example, from Western Africa. We see a small amount of ancestry from East Africa, and also very small amounts of East Asian and Native American ancestry, at least in these particular populations. If you look at populations from other regions, you may see more ancestry from those regions. And again, this is reflecting the history of the transatlantic slave trade originating from West Africa. And actually a very large source of the slave trade was from Angola. And we currently know very little about genetic variation in that region. And that's going to be important to know for some studies in which knowing variation in African ancestral populations will be important for identifying disease risk alleles in African American or Afro-Caribbean populations. I want to tell you about another study that I did with collaborators in which we looked at over 250,000 single nucleotide polymorphisms, or SNPs. These are just regions of the genome that differ at a single nucleotide. And we looked at them predominantly in Western populations along the coast of Africa, and one group from Southern Africa. And when we do this principal component analysis, one of the interesting results is that the distribution really reflects the geography of these populations. And that's not a huge surprise. It means that people who live near each other tend to mate with each other, and people who live further apart are not intermixing as often, and so they tend to be more genetically differentiated. We then did a principal component analysis, including the African American individuals shown here in sort of fuchsia color, and shown in red are Europeans, and then here we have the different West African populations. And we could actually determine the amount of European or African ancestry in any individual, African American individual, by looking at their position along principal component 1. So for example, this individual here, this African American individual, appears to have more European ancestry, whereas this African American individual seems to have more West African ancestry. And then using an approach that was developed by Carlos Bustamante's lab, it was possible to actually scan along chromosomes. So here we're showing the different chromosomes starting at 22, 21, 20, and so on down to chromosome 1. And as you scan along the chromosome in any particular region, you can infer if somebody has African ancestry, which is shown in blue, European ancestry, which is shown in red, or mixed ancestry, which is shown in green. And what we see is that most African Americans have a mixture of ancestry. So they tend to have a lot of, not surprisingly, African ancestry shown in blue. There are regions of mixed ancestry shown in green. But also note that there are some regions of the genome which are only of European ancestry. And this differs quite a bit amongst different individuals. Here's an example of someone who appears to have undergone very recent admixture. They have a lot of African ancestry. Here's someone who has very recent European ancestry. So we see a lot of regions of the genome where they're of mixed ancestry. Here's someone who they self-identify as African American, but they have almost no African ancestry. So that goes to show you that there can be a lot of genetic variation that may not always correlate with self-identified ethnicity. The other important point here is that in the future, there's the ideal that we have is to develop more personalized medicine that is tailored for the individual. And here's someone that, for example, if they went to the doctor and they self-identified as African American, the doctor might prescribe certain drugs that say are more effective in African Americans. But what if at that particular position, where they have only European ancestry, what if there was a drug metabolizing enzyme gene at that particular point? And so that would be a pure European ancestry. And so that might be important to know. So this has important implications for future design of future personalized medical approaches for treatment. So in conclusion, people from different geographic regions are genetically more similar to each other. So for example, Asian individuals will be more similar to other Asian individuals, Europeans more similar to other Europeans. But in Africa, there's been more time to accumulate genetic variation. They've had larger effective population sizes, so they've maintained a lot of variation, and they've lived in diverse environments. So they tend to be highly differentiated from each other, although we also can see that there's been a history of admixture throughout much of Africa. So therefore, Africans have the highest level of genetic variation, both within and between populations. And we saw that African Americans have ancestry from West Africa and Europe, and that the ancestry varies along chromosomes, which has important implications for personalized medicine. And that concludes this portion of my lecture. And for this section, I'd like to acknowledge the many individuals who contributed together with our funding organizations.